Our next presentation will be by Jeff Sanders. Jeff Sanders received his BS in Applied Physics from Worcester Polytechnic Institute. Then he went on to earn a PhD in biophysics and molecular pharmacology from Thomas Jefferson Medical College. Since Jeff joined Schrodinger in 2013, he served several roles in both the scientific and technical aspects of computational chemistry software. He's currently a principal scientist in the Materials Science Division, focusing on physics-based simulations in consumer packaged goods and applications. The title of his talk today will be How Digital Molecular Simulations Will Drive the Next Generation of Innovation in Reformulation and Sustainability of Consumer Packaged Goods. I'm looking very much forward to this talk. Jeff?
Thank you, and thank you everybody for taking the time today. Hopefully by the end of the talk you'll understand what molecular simulation is and how we can use this as a tool to help drive sustainability efforts in various different areas. And I'll focus mainly on formulations, surfactant, oil-based products and packaging, but know that it extends well beyond that. So just for those that are not familiar with Schrodinger, we have a goal of improving human health and quality of life by transforming the way therapeutics and materials are discovered, designed, and formulated. So materials means a lot of things. I focus on consumer goods. Most of us in the audience will know, but personal care, cleaning solution, food and beverage, and everything in between. So our goal is to provide software through a platform that allows you to discover the next best thing. So what is molecular modeling? Well, it's actually a lot of different things, and depending on what properties you're after, what you're interested in, you may use different types or different levels of theory, as we generally say. So most of you are probably familiar with, multiphysics or continuum FEA level simulations of finite element, where you're simulating an entire chemical engineering level process, a whole part or product. We are way smaller and on both time scales and length scales. So we'll go all the way down to 10 to 100 atoms, where we'll use quantum chemistry level of theory to describe important electronic properties, and we'll use density functional theory commonly, which is very important for looking at optical properties and also stability properties, which is very important for ingredient degradation and stability. Moving up to more larger condensed phase simulations, so we're now talking tens to hundreds of thousands, even millions of atoms. We're using Newton's second law, F equals ma, to describe the evolution of those systems, and we're representing the atoms and bonds as balls and springs with fixed charge properties. Polarization is not generally done. Oftentimes, and a really good example is surfactant morphology. We're not on the length scales with all atoms that we can look at phase separation or assembly of bicontinuous emulsions or micelle morphology. So we'll move to coarse graining, where now we're representing multiple atoms as a bead, and we're parameterizing it depending on what type of coarse grain simulation we're running to represent the most relevant physics and non-bonded and bonded interactions without losing too much accuracy, while able to speed up the simulation. And since I haven't explicitly mentioned time, often the atomistic simulations are on the nanoseconds to a few microseconds, and the mesoscale simulations are on the microseconds up to milliseconds, depending on the level of coarse graining. Molecular simulation has been around for a long time. There was a Nobel Prize that was awarded the year I left grad school. So it's something that has clearly been established, has very large impact in the scientific community when trying to understand the molecular details of your system. And for a long time, it required a PhD in this area or a related area, and everything was done, via command line. So you see, like in movies with hackers and a bunch of text program. We wanted to change that by providing an interface that is unified to use all of the tools I just mentioned, but within a single application platform. And that is required years and years of development, hundreds of developers. But now we provide a tool that allows you to run various levels of simulation, take output from one to another, and do this without having to learn a programming language or have to understand, get a PhD in statistical physics. So lowering the barrier so that everyone can run simulations, because there's a lot of insight and a lot of multidisciplinary areas like consumer goods, food and beverage, oils, surfactants, personal care, where understanding and bringing a different insight to modeling is very useful. So when we talk about modeling, I mentioned a lot of physics-based modeling using quantum, classical mechanics, and coarse graining. There are other ways, and so we also focus on machine learning, understanding how we can leverage all the data, either through experiment that already exists in-house, or perhaps generating data using physics-based simulations that is predictive, and then using that to build a machine learned model. And then, of course, tying that all together at an enterprise level so that experimentalists, engineers, modelers, and even management can see the data in real time and then ideate over this. So we think of one example, one formulation, we run some simulations, we get some data back. Maybe someone wants to change a composition or add a new molecule. So being able to do this, generate the data in a short amount of time, and then share this data at an organization-wide level, democratizing simulation, is something that we at Schrodinger focus on heavily. And the question is, why now? Why do we care about digital chemistry in general? Why do we care about modeling, right? Well, there's a changing demand, sustainability. I could go on, and I think everyone knows and understands why we can't keep using and consuming plastics the way we do. The economic toll that traditional animal-based meats have on our environment. We need to think about how we can revolutionize and change so that we still have a world that's livable in 50 years. And so this is one drive, right? And this is also leveraged on, we need to be able to run these simulations and use computational power. And so really our contributions is to provide these tools, as I mentioned earlier, in a way that's accessible to experts and non-experts alike, and also distributed on a much higher level. And so, since I've mentioned sustainability, there are a lot of challenges, not just in surfactant-based applications coming up with new plant-based surfactants, or trying to replace one that maybe consumers no longer want to see in their product, but also the packaging itself, the plastics, the papers that we use that our product goes in, either going to transport to another business to business or going direct to consumer. And so there are a lot of challenges associated with this. Plastics are a big one, impact both from an economic, environmental standpoint. How do we drop in replacements for traditional components? That's a very difficult problem. We can't go out today and replace PET or PE or polypropylene PP with something else, a bio-based polysaccharide polymer. It doesn't work that easily. So we really need to address and understand, as we replace and improve and generate new packaging materials or new formulations for products, how difficult is this modeling be used to lower that barrier? And this is, of course, driven by a lot of global initiatives, changes from the USDA, from EU, for what can and can't go in our foods, can't go in our cleaning agents, can't go in our packages. So there's serious motivation to do this, and to do this now. So the idea and the goal of doing this, right, is we take a sustainable or renewable feedstock, which plants are everywhere, right? Large biomass, and they can generate or be used to generate a lot of useful building blocks, fatty acids, terpenes, flavor molecules, amino acids, other proteins in general. So can we get those? Can we convert them, or can we separate them? And then can we build a product that then can be used, and when it's done and it's discarded, it can be degraded through some biological mechanism? And then can what it's degraded into not be toxic, but be used as like CO2 or water or other organic or inorganic degradation products that can then be fed back into a renewable feedstock? So stepping away from sustainability for a second and focusing on trying to make new materials, new chemicals in general, it's a tough process. It takes 10 to 20 years. It's slow. A lot of it is hit or miss or trial and error. You think of the dartboard analogy when you're making a new formulation. That's not very rigorous. It can be very, expensive, and you can end up with no new product. So why not we try and use a tool that allows us to spend less time, spend less money, get additional insight into what we're trying to develop or redevelop. And so when we think about this, we can use modeling in two different lights. We can use it for continued development, understanding how formulations are rationalize the way a product you've used for 50 years behaves, or we can use it to design new chemistries or new formulations, new packaging products. And this is faster and it's cheaper, and there's a lot of advantages. So let's focus, now that we've set the stage here on molecular modeling of surfactant-based solutions and how you could use this when coming up with new sustainable surfactants. So these are just a very few examples, and I'll illustrate, what can modeling tell us with surfactants. It can tell us a lot of things. Some of the main things that we're interested in are usually morphology, and how we can use surfactants to model behaviors of different temperature, different pH, different salt conditions. So we can look at, phase diagrams of surfactants and maybe a solvent, a co-solvent in water, and we're able to model very quickly how we can form very large, assemblies, things like liposomes, mixed micelles, rod-like. You can go through the entire phase diagram, and you can do this with coarse grain simulation. And it's been done in the literature for many, many years. And there are a lot of great successes in understanding not just simple surfactants, but mixed formulations of surfactants, adding in flavor molecules and adding in important things like maybe nutrient precursors. So this is a more relevant, more tractable example where maybe we want to get more, beta carotene, right, essential vitamins into a new beverage. So we need to create an emulsion, so we need some type of surfactant. Tween 80 is the commercial surfactant, right? It's something we may want to replace in the future, but if we want to understand how does it emulsify beta carotene, is it stable? If I change something in that beverage, will it stay the same, or will it crash out? So looking at a simple modeling, we can throw all of these in a simulation cell with water, and we can look at how does an emulsion begin? What's the size of the emulsion? If I heat it up, what happens to that emulsion? If I add more salts. And so this is just a very, very simple example. We're looking at, okay, we put these, surfactants, we allow them to interact, and how are they able to form stable emulsions? So we can understand when we add more things, what happens. We can also take it a step further, right? So if we look at biosurfactants or more green surfactants, and we can look at oil emulsification, we can very quickly run these simulations, and this can be done in probably a few hours, and we can look at how quickly can a particular biosurfactant emulsify an oil. This is, again, using the same sets of simulation techniques, but now we're thinking about changing maybe tween to something else. And as we add more and more simulation studies, and as we understand and add more components, we can get a sense of what does the morphology look like. Morphology isn't the only thing, though. We also want to understand some of the critical, properties we can measure in the lab, like critical micelle concentration, viscosity, or morphology. And so we're able to do this with some of our simulation workflows and predict these properties. Maybe we're able to predict them quantitatively, or maybe we're able to rank order, or maybe we're able to tell you whether it's getting better or worse or staying the same. Traditional small molecular weight surfactants are what we usually see. Those are the nice things I can show you. But as we move towards more plant and natural sourced ingredients, we need to think about proteins and polysaccharides, right? Borrowing from nature to do these things. Proteins and polysaccharides are really good at effective emulsifying an oil. There are many different types. You can mix them together. They can form very complex interfaces between oil and water, and understanding these, whether the protein partially unfolds, how the polysaccharide interacts is very important. And so this is something that is not very easy to study experimentally. If you want to look at how are these interacting, you're looking at either bulk level properties, but you're not going to get very high resolution structures. And they're, as a result of that, they are less well known than traditional synthetic, low molecular weight surfactants. And as we add more and more complexity, we need to understand, can these polysaccharides, can these proteins do the same job as traditional surfactants, and how are they able to do that? And that insight allows us to better iterate or design new emulsions using bio-based, particles or molecules. And then, to show a very simple example from nature, there are very surface active proteins called hydrophobins that exist within fungi. They're very important for the development cycle and reproductive cycle. And they're they're actually the most surface active proteins in nature. And we know what they look like from a structural perspective. It's a little harder to draw a protein than it is a small surfactant, but we know what the crystal structures look like, and we have an understanding of what are the types of assembly modes, whether you're at an interface or whether you're in solution, whether you can form a dimer, a polymer, an air-water interface is a self-assembled monolayer, or a fiber, which may be important for plant-based protein applications like alternative meats. So we can take these structures, we can look at oil-water interfaces, we can look at self-assembly, we can look at surface absorption. This is a whole other talk if I wanted to dive into it, but just one simple example is we can, again, simulate with water, and I'm always hiding water for the most part because it just gets in the way from all the other pretty colors, to look at if we simulate maybe a few of these proteins, the hydrophobins in a box, and understanding, how they generally assemble. Maybe the green part is what's known about how they form a dimer. What happens when you put more of them in there? You drive up the concentration to form a fibril. And so this is something that's very hard to do experimentally, to understand what these fibers look like, and you need to understand what the non-covalent interactions that are driving assembly if you want to better tailor them to serve a particular function in your products. And then additionally, beyond just using them as an emulsifying agent, you might also want to use them to encapsulate something. So encapsulation is important for a lot of reasons. Food and beverage are a really good example, or fragrance. And polysaccharides specifically, and some proteins, are very good at encapsulating molecules so that they don't degrade, they don't interact with things that they shouldn't until the product is put into use, whether you're eating it, whether you're spraying it, or whether you're mixing it into something else. And it's been known for a long time that starch-like molecules, amylose being a good example, can encapsulate, or it can bind to lipids and other small hydrophobic molecules. If we want to understand how to simulate those, we want to make sure that we can simulate the properties for polysaccharides correctly. And so we're going from understanding the chemistry to parameterizing a particular structure, like a small chain or a medium chain amylose molecule, nice represented polysaccharide. And can we predict relevant physical properties of just the polysaccharide itself before we go do more complicated things, like look at encapsulation? And it turns out we can predict moisture activity, so moisture uptake is a function of relative humidity or temperature, and we can accurately model with an offset the plasticization effects of water on starch, amorphous starch systems. And this is a paper we published last year where we get really nice agreement with just simulations alone with all the data that's available in the starch technology textbook. So back to encapsulation, we're thinking about how do we, make inclusion complexes with, surfactants, other ingredients that we don't want to interact with solvent or otherwise, and how do we quantify that, right? So in traditional, host guest binding or protein ligand, or however you want to call it, we can thermodynamically describe these interactions and the stability. And so we can do that, using Gibbs free energy. We can calculate what's, if we know the binding affinity for a molecule and maybe a starch, an individual starch chain, we can calculate using simulations what is the relative binding constant, and also what's the free energy associated with that. So if we're selecting a particular set of molecules or understanding competition between other molecules, we can rank order these and we can get the right trends. And a really good example here is looking at, we want to calculate the binding as we increase like an alkane chain for particular type of flavor molecules. Are we getting the right trends? And it turns out, we compare against some, experiments that have been done in the literature, we're actually getting, an increase or decreased binding strength as a function of the alkane chain length. So, key point here is that, longer linear chains form more stable complexes. That might be something we could infer looking at the structure itself and some experimental information, but we're able to not only explicitly model this, but we're also able to quantify those interactions, rank order them, and they agree with experiment. And for the last thing I'm going to talk about, I know I'm running close on the clock here, is the packaging side, right? So that's just formulating on the product side, but what about packaging? And a really good example that we like to show is BPA, because everybody knows to buy a BPA-free bottle. It's been something that's been talked about in the news, media, and something I think almost everybody's aware of when trying to buy your next back seal water bottle. Is it BPA-free, right? Because BPA is not great if you're consuming a lot of it, but it is also a contaminant and something that exists in polycarbonate polymers. So if we look at a plastic that might be a color-coded bottle, right? So it has a dye, it has BPA, and it has a polymer. How does that BPA leach into solution? And so maybe we're looking at a high ABV beverage. You can pick other ones, and we're going to model the interface, right? So when does the right where the product touches your coating, your packaging, or your first layer? What does that look like? What happens when you heat it up? Is BPA able to leach into solution, which it is in this particular case as we go from left to right, increasing temperature. We can see our little green BPA molecules are moving into solution as we do that. So this is a good way to qualitatively say, okay, at room temperature, it's probably all right. We're not getting much BPA into our high ABV liquor. But as we increase the temperature, maybe it sits on the truck, maybe it has to go through processing, maybe it's just a hot day in the warehouse, we're going to have more BPA leaching. And we can go a step further from that and actually quantify it. So we can look at what's the diffusion and what's the diffusion into the solution versus diffusion within the polymer matrix. And again, this is all something that we can easily do and analyze. Last, and I'll end on this, is looking at more bio-based packaging for the future. So looking at replacing traditional plastics, as I mentioned earlier, with things like chitosan, chitin, or cellulose, and then polylactic acid, right? So things that are renewable, they're thermoplastic polyesters. They need to be mixed. These are very hard to create an equivalent plastic, but we need good thermomechanical properties for a blend in order to create a drop-in replacement for the plastic bottles we use today. And so one of my colleagues in Europe has been working on understanding not only how do these mixtures give you insight. First, sorry, the slide is jumping around. How do we identify these things properly? Are we packing them? How do they phase separate? What does it look like if we have a particular polysaccharide or long chain biopolymer and PLA? How do they phase separate on the nanoscale? Understanding that. Understanding how they might create pores if there is some phase separation, quantifying all of that on a per chemistry basis. And then also understanding, do we get the right thermomechanical properties, the real things that we're after? So we get the TG trends, right? So as we add increasing polysaccharide content, we see that the TG goes up. That agrees with the experiment. And also mechanical properties. So as we add more single polysaccharides, we see that, both uniaxial tension and shear tension increase. So our thermomechanical property predictions are in line with experiment. We may not get the exact same number, but we're getting the trends right. And then also water loading, right? Moisture, which is really bad for product stability. Can we load the water in there? And where does the water go? Does it go near in the cellulose, or does it go to the interface between the PLA and the cellulose? And so understanding, not just can we predict relevant thermomechanical properties and moisture uptake, but getting the molecular insight so that we can help better inform decisions on how we design these products moving forward. And then, of course, plasticization effects on TG or stress strain properties, mechanical properties are all things we're doing, and we're working on a publication for this. So just to round out, I know that was a lot. I try to cover a fair, broad spectrum because I know the audience is very diverse. But takeaways, right? What can we get out of this? So molecular modeling can be used to explore formulations and also reformulations. I tend to think reformulating is just formulations, but depends on who you talk to. You can understand morphology, environmental effects, which can lead into product stability or shelf life. And you can model not just the product itself, but the packaging. Sustainability is not going away. It's going to require novel formulations of product designs. It could be very expensive if you do experimentally or if you're just doing trial and error. But if you're using simulations as a way to triage good versus bad ideas, it's a lot cheaper and a lot faster. modeling is never going to replace experiments, but it can enhance them, and it can be related either indirectly or directly to experiments. I'm just showing a bunch of examples where we do agree with experiment in varying degrees, which allows you to either narrow your search space, whatever that may be, chemical space, formulation, or packaging space. And by reducing these numbers of experiments, right, we're also reducing and increasing sustainability from an organization standpoint by, using less energy, using less water, using less expensive raw materials, and less manpower to get results that aren't bench level experiments. And to end, does this work? Yes, it does, and not just for us, but our customers use it. And there's a really good blog on our external collaboration page, where one of our customers, Reckitt, talks about how they were able to adopt simulation. They were not simulation experts. They were able to get useful data in a few months. And for them, they've seen on average that they're able to expedite timelines 10x compared to just using experiment alone. So not only are you able to drop in, replace simulations for some of your experiments, and that itself is sustainable, but you're also increasing and accelerating discovery of new packaging and new formulations. And on that, I will say thank you, and I'm happy to take any questions.
Thank you, Jeff. A fascinating topic and obviously very powerful. I had a couple questions here. The first question, do you see that the people that use this technology are able to populate your data set with what they know and then run some experiments through your Schrodinger platform and then use the results of that to inform their next steps for lab research, and then kind of just weave back and forth like that?
Yes. So we call that an ideation cycle, where you or I come up with an idea, we hypothesis test it with simulation, we run a bunch, maybe 20 or 50 formulations or 10 different packaging materials. We decide which ones might be best or worst, depend on a single property or multiple properties. Go into the lab, instead of making all 10, now we'll just make 5 or 3 if we, depending on how the data spread looks. And then that experiment might inform, okay, this is good, but it's not quite good enough. You're going to go back to simulation. You're going to refine those simulations to further narrow the scope. So that's the primary way we see doing it. The other option is if you have some experimental information and you've generated some physics-based simulation, we can use tools from the machine learning realm, things like active learning, that will do this iteration and will combine multiple types of data to build a model. Of course, you need enough data to do that, but if you predict a lot of simulation and you have some experiment, you may be able to allow that to churn out some ideas itself, rather than just having to, do it the old-fashioned way of simulate, experiment, refine, and then, iterate again.
Very, very interesting. Lastly, the question is, what areas of opportunity do you see that are most excited about your technology?
So I don't know that I would name only one, but where I see the technology going is to really help aid novel discovery. And what I mean by novel discovery is, we can take formulations, we can take a wealth of insight from an organization and say, we need to change a product a little bit. We know what works, what doesn't work. That intuition is great and is necessary, but sometimes, depending on where that intuition comes from, where that wealth of knowledge comes from, may cause you to miss a key new novel molecule, formulation, packaging, or interface, or you might miss something. So where I think this is going to get really exciting is it's going to take people in directions they didn't think that were possible, or that their knowledge, not necessarily saying that their intuition is wrong because they've been doing this way longer than I have or someone else, but it may open up new avenues that people didn't think to explore or couldn't explore because just running the experiments is very expensive. So I think modeling, to me, is the most exciting. Is it's going to push us in new areas we didn't think we could go before in chemical space, and it's not going to have to rely on some of the old, is this cheap enough that we can try doing it first?
That's very exciting. And this is where scientists really start to, look around and get really excited when they see that there's a hint that you could apply something else to this problem that I've been working on, an idea that they never had in application. Thank you very much. I appreciate your time today. And this concludes our webinar. It will be available live or available online later. And we invite you to participate in our AOCS technology sessions and the upcoming AOCS annual meeting.